It's all yours.
Okay, thank you, Lydia. I appreciate that.
And how do I turn this off, Kelly? I think I
Okay, so what I'm going to do today is talk about one of my favorite subjects, which is CMV. And since I have a very clinical perspective on clinical pathology, I still practice infectious diseases, what I thought I would do is kind of take you through my two worlds. And I'm going to take you through a validation of a laboratory-developed viral load assay for CMV, so you can appreciate the laboratory side of it. Then I'm going to take you through the clinical side of it, of how physicians actually use these tests to manage patients, and how understanding the laboratory performance really impacts how clinicians should use the test. And that's really where the pathology, clinical pathology side of it comes in, is helping us, our need to educate clinicians of how to use tests. And I'm supposed to disclose, these are scientific advisory boards that I've sat on in the last couple of years, and we're supposed to be starting a small business grant with a company called IQ in the next couple of months. Okay, so let me give you some background on Cytomegalovirus. It's one of the herpes viruses, so it's an envelope double-stranded DNA virus, and it is a very common virus in the population. Healthy children, about 10 to 30% of kids are infected, and about 70 to 40% of adults are infected. And your risk really, your likelihood of being infected depends on a variety of risk factors that you might have. Sorry, this seems loud. It's loud to use, it is to me. And high times of getting infected in your life are when you're a young child in a daycare center and you're slobbering all over your friends. The virus can be in saliva, urine. So that's a great time to get infected. And another time to get infected is at the time of sexual activity. So most adults who are going to be infected are usually infected by the time they're 30 or 40 years old. Now, what are the common manifestations? Usually it's completely asymptomatic. If you're a healthy child or a healthy young adult, you have no, likely you'll have no symptoms whatsoever when you get infected. The most common symptoms when they occur in immunocompetent people is a mononucleosis type syndrome. So it's really indistinguishable from an EBV type mono. Then you develop a lifelong latent infection, and as long as you remain healthy, this virus will do very little. You will excrete it sometimes in your urine or in your respiratory secretions if you get really stressed out. So if you probably cultured PAC residents just before the boards, I'm sure a portion of them would be able to grow CMV from their urine. But from a disease perspective, you have no problems with it. It's only once you become immunocompromised that you then reactivate the virus. We're not going to talk at all today about congenital CMV, but that's a whole issue in itself, getting your primary infection during pregnancy. So the way that virus presents is really driven by a lot of different factors. And one of the reasons that CMV is so interesting to me is it's very complicated both from a laboratory perspective and from a clinical perspective. So how you manifest a CMV infection if you're immunocompromised depends on a lot of different factors, and one is the type of transplant. So stem cell transplant recipients tend to have severe disease when they get it or have the potential to have severe disease. Solid organ transplants, it goes a gamut. Kidney transplant recipients tend to have less severe disease than lung transplant recipients. And so that's probably driven in some degree by the level of immunosuppression that the various organs require. So if you're doing well with the kidney transplant, you're going to have a lower risk of infection than if you're doing well with the lung transplant because the amount of immunosuppression involved is different. Now what's also key is the serostatus of the recipient and the donor. So if I'm seronegative and I get a seropositive donor, that means I'm going to get my primary infection when I'm immunocompromised. And those people tend to get very sick. And they tend to be treated, managed differently post-transplant than if I'm already seropositive and then I get a seropositive organ. And then finally, the degree of immunosuppression. So even within, say, kidney transplant recipients, if you're doing well and you're on a relatively low amount of immunosuppression, your risk of CMV reactivation is lower than if you keep having episodes of rejection and you have higher levels of immunosuppression. You're going to end up with a higher risk. So a lot involved. The way patients present is also very diverse. Some people just present with some fatigue and fever. Other people present with quite severe disease like pneumonitis, which we see more commonly in stem cell transplants. a variety of GI disease, starting in the esophagus, going all the way down to the colon. And central nervous system disease occurs. It is not as common, I think, as it is when we were at the peak of the HIV epidemic and we didn't have good antiretroviral therapy. We tended to see more CMV disease in the neurologic disease in HIV patients than we do in transplant patients. And the management is fun because it depends on multiple clinical and laboratory factors. So let me give you a few definitions that I'm going to use. We talked about latent infection. Now, CMV infection is defined usually by a positive laboratory test. So you either culture the virus from blood or some sort of body fluid, but the patient is completely asymptomatic. And now that we've moved to molecular testing, it may be that all you have is a positive PCR, the patient is completely asymptomatic. So that's infection. You know either the virus or the DNA is there, but you don't have any symptoms associated. Where disease really is you can detect the virus and there's some sort of clinical symptoms or end organ dysfunction. Now, how do we manage patients? A couple of terms I want to throw out because some of the studies I'm going to take you through will use these. Prophylactic therapy is when you basically treat all patients in a certain group. Okay, so for instance, anybody who's seronegative, who gets a seropositive donor, their risk of active disease is extremely high. So those people will all get treated with ganciclovir or valganciclovir. The goal is to prevent the development of disease. And prophylactic therapy is extremely effective while the patient remains on therapy. It's also been shown in solid organ transplant recipients to reduce bacterial and fungal infections, and this is because CMV is an immune-modulating virus, so in itself it can be an increase the immunocompromised state. So it increases risks of other types of infections. Once you stop your prophylactic therapy, then you run the risk of getting CMV infections down the line. So we may prophylax people for 3 months or 6 months, and so when you stop the prophylactic therapy, you have to be careful because you can then develop CMV disease later. This works, but there's a little downside to it. One is the drugs that we use to treat CMV are quite toxic, so you have that problem. Plus, they're very expensive, and so it works on one hand, but it has the downsides on the other hand. Another term that we use a lot is preemptive therapy, and that is instead of treating the whole group, you do some sort of laboratory test to try and find the high-risk patients and then only treat those. And this is a strategy that's used in many transplant centers now, where we monitor people after transplant with a viral load assay, and when they get a certain threshold viral load, then we'll give them therapy even if they're without symptoms, because we think they're at the very high risk of getting disease if we don't do that. And I will show you some clinical data later to support that. So how do we treat people? We talked about prophylactic versus preemptive therapy. The main agent that we use is either IV ganciclovir if the patient can't absorb drug yet, or oral valganciclovir. Foscarnet and cidovir are also used, but they're second-line therapies, and we also can use immunoglobulin in severe infections. And like I said, I won't go into this now because it's really not a primary interest for us today, but each of these drugs has considerable toxicity associated with it. Okay, so what are the available viral load tests out there if you're going to monitor your patients for disease? And the pickings are pretty slim. There's this hybrid capture assay out there by Digene that measures CMV DNA in whole blood. It was FDA cleared years ago as a qualitative test, and then many labs have modified it to be quantitative. It's not very sensitive. It only goes down to about 1,400 copies per mil. And it's a very tedious, labor-intensive assay, and it's slowly but surely kind of being phased out of the market. There are very few labs that still use this. There's the Ampicor Monitor CMV test. This is a conventional PCR assay that Roche developed but never put through the FDA. And laboratories have used that in the past, but most labs, again, have moved away from this and are using real-time PCR assays. And anybody that's using a real-time PCR assay in this country right now has basically developed it themselves. And you can either develop it completely from scratch or you can use analyte-specific reagents, which are primers and probes and reagents that you can buy that are manufactured under good manufacturing practices, and then you mix them together and develop your assay. And I would say if you look at the CAP proficiency testing, that 80% of labs in this country are using some sort of laboratory-developed test. So you're going to develop a test. Okay, that's good. That's an important thing. CMV is an important disease. You should have the test. How are you going to go about designing that? How are you going to think that through? Well, the first thing we want to test it sensitive. Now, I don't want it to be too sensitive because I don't want to detect latent virus. So we have to balance that sensitivity. And CMV PCR has been on a roller coaster ride over the years because in the early days of PCR, back in the mid-90s to late 90s, there were always reports out about how incredibly sensitive CMV PCR was. And one of them that came out was in seronegative blood donors, completely healthy seronegative blood donors, that 20% of people were positive. And their conclusion was, wow, look how sensitive this assay is. Those of us that did molecular testing at the time said, oh my God, you idiot, you contaminated your laboratory. And it turns out that, you really can't have DNA present unless you're seropositive, unless there's something wrong with the test that you're running. So you have to be careful about sensitivity. You don't want it to be so sensitive, and you don't want to also contaminate your lab. You need to test it specific. You don't want to detect the other herpes viruses with the assay. Linear range is clinically important, and we'll go through what a clinically important linear range is. And then you want an assay that's reproducible and an assay that's accurate. Now, most of us have moved to real-time technology. I'm going to take you through just a few slides so that the data that I show you subsequent you'll be able to put into a context. In the old days, we used to do a nucleic acid extraction, and then we would do an amplification, and then we would do a detection step. And that detection step could be running it on a gel. We then moved from that to oligo-based hybridization assays, and we finally now have moved to the next step, which is amplification and detection occur simultaneously. Okay, so while you're putting your primers into your PCR assay, you also put a hydrolyzable probe in there. And so while you're amplifying, you're also detecting. And what you do is you measure fluorescence. And this is what the assay is, how they're designed. And this is just the Pac-Man chemistry. There are many other types of chemistries, but we don't have time to go through them all today. But this is your target. So this is our CMV target. We bind a specific primer. And then the polymerase, the Taq polymerase binds to your primer and starts synthesizing DNA. And also in the reaction mixture is a probe. The probe hybridizes to an area inside of your two primers. And on one end of the probe is a reporter fluorescent molecule, and on the other end is a quencher. So if you excite this, it will fluoresce, but it will be quenched by the quencher molecule on the other end of the probe. Okay? So now, one of the features of Taq polymerase is that it has a 5' exonuclease activity. So as the primer lays down and you start synthesizing the next strand of DNA, once the enzyme actually bumps into the probe, it's at the 5' end of that probe. And so what does it do? It starts hydrolyzing the probe. And once the probe is hydrolyzed, the reporter molecule floats away from the quencher molecule, and you can measure fluorescence. And that's exactly what the plots look like. So this is cycle number down here, and this is fluorescence that you're measuring here. And this is a low level of fluorescence, and then all of a sudden you get to a threshold, and you just have exponential amplification and fluorescence detection here. And then you set a certain threshold that once you cross that threshold, that's a positive result. And what you're going to hear me talk about are CT values, which are crossing thresholds. And so it is the cycle number at which the amplification crosses the threshold, which in this case is somewhere around 29. If you have less virus in the sample, it's going to cross way out here because it's going to take more time. And what real-time PCR has revolutionized for us in the laboratory is quantification. Because now you can plot your concentration of your molecule versus the crossing point. And you have an inverse relationship. So the higher the concentration, the lower the crossing point. And you can plot, on a log plot, you can plot a linear line and use that to calculate the concentration of virus in a sample. So I would put on a variety of standards. I would figure out what the CT is for each of those standards at a known concentration. I get my standard curve, and then I'll know the CT value of my patient, and it will allow me to calculate the viral load. So, if we're going to go around to develop, and I'm going to take you through an assay that we actually developed in the laboratory, and it's real live data. I didn't make it up. So you'll see it in all of its ugliness as it really happens. And so we decided that we were going to develop this assay, and we ended up using primers and probes that were supplied as an analyte-specific reagent. You didn't have to do that. You could have a chemical facility manufacture those for you. But we have just found over the years that the quality control is a lot easier if you use these analyte-specific reagents. Common targets are the polymerase gene, the major immediate early antigen, and the glycoprotein B. We actually have targeted this gene right here. The assay has an internal control, which is extremely important because this is a DNA that is added to the specimen prior to extraction, and then you detect it at the end of the assay, and if that molecule isn't amplified, then you know that something went wrong with your assay. Either the amplification failed, or there's an inhibitor of amplification in the clinical specimen. So it's very important to be able to add these internal controls to your sample. Okay, so the way we put ours together was to build a standard curve with each run. You don't have to do this. You can generate a standard curve over, say, a dozen runs, and then just determine what your mean slope and intercept is and use that to calculate viral loads. But our viral load testing volume is so high for CMV that it's not a cost issue for us to include a standard curve each run. We did an automatic nucleic acid extraction method. What we use in the laboratory, our workhorse test, is the MagnaPure. A lot of the data that I'm going to show you today was actually done with a manual QIAGEN extraction. And then we determined the limit of detection, the linear range, and the precision of the assay. These are all things that you need to determine before you offer an assay clinically, because if you don't understand the performance of the assay and you haven't validated the assay, you actually can't bring it up live and run it. We purchased standard curve material that had a very high concentration, 1 million copies per mil, and then we did dilutions down to about 25 copies per mil to figure out what the limit of detection is in the linear range. So this is the limit of detection data, and this is with plasma samples. And so this is the concentration, and I have log here, and for those of you that don't convert instantly in your head, I put the copies numbers here also. This is the number of replicates that we tested and the number that were positive, and then this is the percent positive. And so what you can see, at 1,000 copies, we had 100% sensitivity. At 200 copies, we had 95%. At 100 copies, we had 90%. At 50 copies, we got it 69% of the time. Now, there's actually a definition of limit of detection out there, and it's the one that the FDA uses, and that is that 95% of your replicates need to be positive in order, at that point, and that's your limit of detection. So, our limit of detection for this assay is about 200 copies per mil. Okay? Now, this is the linear range of the assay, and you can see it's not a bad linear range. The R squared is about 0.99, and it's from 100 copies down to up to 1 million copies is linear. Actually, it is linear up to 10 million, but at the time we did the initial validation, we couldn't find material with a viral load of greater than 1 million. So once we brought the assay live, a couple years later, we got a young patient, a child, who had a viral load of over 10 million, and so we were able to go back and do serodilutions and show that the assay was linear up to 10 million. Here's the reproducibility data. This will make the clinical chemists in the audience absolutely gasp. But this is our reality in molecular microbiology. So this is the concentration that we tested, and this is testing over multiple runs on multiple days. And the standard deviation is here, and the percent CV is here. So when I first went to Emory, I was shared office space with one of our clinical chemists. And I said, Jim, what's the CV on blood glucose, or he goes, or cholesterol or something? He goes, oh, cholesterol is one of our worst tests. It's a terrible coefficient of variation. It's like a couple percent. And I'm like, oh my God. I said, what would you think about a CV of 50%? And he said, oh my God, are you kidding me? And that is what molecular diagnostics is. Now remember, I'm measuring extraordinarily low amounts of material in the blood, multiple logs lower than you measure in a glucose assay. So that's how I defend myself. But what's interesting and what you need to understand about these standard deviations is a standard deviation of 0.15 log is needed to distinguish 5-fold differences. This has been shown for HIV viral load. So if you don't have a standard deviation at 0.15 or less, that means you can't tell 5-fold differences apart. And this is really, really important because clinicians think a difference between 100 copies per mil and 200 copies per mil is important. And your job is to explain to them that, in fact, no, it is not. And one of the best, most effective ways to prevent clinicians from over-interpreting small changes in viral load is to log transform the data and then tell them that anything less than about a half a log is within the variability of the assay, or anything less than maybe 0.3 log is within the variability of the assay. And it helps train their minds to what important differences are. I will tell you that most clinicians do not understand logs, and so we report our data as copies per mil and also log copies per mil so that they can begin to understand how they go back and forth from each other. But you can tell from these standard deviations that we are not going to be able to reliably tell small differences in viral load values. Now let's talk about accuracy, because for accuracy you need to know what truth is. And one of the biggest problems we have in molecular microbiology is we don't know truth. There is no standard material for CMV that is recognized and agreed upon in an international way. We have international standards for hepatitis C and for HIV, but we don't have anything for CMV. And so when I develop my in-house assay, I pick something as standard curve material. I might grow the virus, I might construct a plasmid, I might buy DNA. If Chris is going to develop an assay, he may choose something different for his standard curve material. And so our values are not necessarily going to agree. And so in the early days of CMV viral load testing, we were kind of struggling. We didn't have very reproducible assays and we didn't have accurate assays. Now we have quite reproducible assays, but nobody knows if their assay is accurate because there's no international standard material that we can all use to calibrate our tests. And this is probably one of the biggest limitations of CMV viral load testing right now. So we don't know who is accurate. And I want to show you data. I'm going to digress just for a second. I want to show you data from a multi-center international study that we participated in to try and get a feel for what's going on with CMV viral load assays out there in the real world. So we built a panel of 12 specimens. 12 of them, 2 of them were negative, and 7 of them were dilutions of purified nucleocapsids. So they grew up nucleocapsids, they purified them, and then they diluted them in plasma. And then we also had 3 clinical specimens that we diluted to see how people did with clinical specimens. There were 33 labs that participated in the U.S., in Canada, and in Europe. And this is what you get. On this axis is the sample number. Okay? On this is the concentration. So these are 2 negative samples right here. We expected them to be negative, and you can see each one had a laboratory that got a false positive result, which isn't too bad out of 33 labs. You'd like it to be better, but that's not horrible. These are, each of these diamonds is a positive result, okay? And these are the nucleocapsid specimens, and these are the clinical specimens. So let's just pick one. So let's pick sample number 8. Somebody got a negative, even though we thought it had a concentration of 1,000. That was based on the nucleocapsids and having several labs tested ahead of time. That's what we thought we were sending out to labs. So one lab down here got a copy number of about 1 log, and this lab up here got a copy number of about 4 log. So there's a 3 log spread on that sample. And if you look at every one of these samples, look at the spread. Look at the spread. It's 3, 2.5 logs spread on every one of those tests. And this is the reality. So Chris can get a viral load of 1,000, and I can get a viral load of 100,000 on the same specimen. And this is driven in large part because we don't have an international standard. Yes?
Is that lab specific, so the ones who are low are low for all the samples?
That's a great question. If you develop a good assay, it is lab specific. If you don't know what you're doing, and I didn't put that curve in there, you don't get that consistency. But most good labs can develop an assay, and I tell you that we work consistently and reproducibly at the low end of this curve. And just so we're reproducibly at the low end of the curve, I'm okay with that. Okay, so why don't these values agree? Well, one is the standard. It's very important to understand that we all use different materials. Some of us use nucleic acid, some of us use culture material, and that how you quantify that material is also important. And so we're all using standard curve material that's completely different. Primers and probes have different efficiencies of amplification. We know this, so that can be a problem. Some labs use different extraction methods, and that can also impact the efficiency of your recovery. Now, so measuring accuracy is not possible right now for CMV. We don't know what truth is. For things, like I said, for like Hep C and Hep B and HIV, where we do have international standards, what happens is the WHO makes these standards, and then, or they contract out to a company that makes these standards, and that becomes truth. And companies then purchase that material and make secondary standards or make calibrators, and those are the calibrators that are used in clinical tests. So if you look at the CAP proficiency for hepatitis C and HIV, the standard deviations between labs is actually pretty tight in a context, of course. But if you look at it for CMV, it's 5-6 times higher because we don't have that international standard. So this is a real problem for us that we need to be working through, and I'll tell you at the end what the plan is. So the last thing that you need to do when you're changing assays or bringing in a new assay is how does your assay compare to the one you've been using? So for instance, right now you're sending out your CMV viral loads, and let's say you develop your own assay and you bring it in-house. One thing that the clinicians are absolutely going to need to know is, are the values the same? Okay? Because that's going to majorly impact their management. So the way to do that is with a bland alternate plot. And on this axis is the average viral load between the two samples. So you do, here I think we get about 70 specimens. You run them with both assays. You take the average of those two values, and then on the y-axis you put the difference between those two values. So for instance, this point here has an average viral load when you take the two tests together of 3, and the difference is 1 log, which means one lab got a viral load of 2 logs, and the other lab the other test gave you a viral load of 4 logs. When you average them together, you get, or that's probably not right, it's 1.5 and 3.5. When you average them together, you get 2, and the difference between them is 1. That isn't quite right, but you get my point. Same thing here. These two assays absolutely, these two tests, samples absolutely agree in the different assays. The difference is 0. Okay? But you see, the average difference in all of these specimens that were tested is very, very low, 0.01 log. Okay, so essentially, on average, they're the same. But look at for any given point. It might be a log down here. Look at this one down here. It's 2 logs. And so you can tell the physicians, on average, these values are going to be the same. But you have to be prepared to run specimens for people who are on therapy. So if my patient's happens to be on therapy when you guys decide to change your assay, I'm going to ask you to go back into the freezer and get an aliquot of that specimen and retest it so that I make sure that for my patient those values agree. And that is extremely difficult, extremely important for clinicians to be able to properly manage their patients. So this is our viral load test. The limit of detection is 2.3 logs or 200 copies. We have, I didn't show the data, but it's specific. There was no cross-reaction with other herpes virus. It's linear between 100 and 1 million copies. Appropriate reproducibility, and we had good agreement with the Amplicor viral load test. So we were ready to go live. Okay? So now the question is, here's my test. I think it's beautiful. How are they going to use it clinically? And what do I need to tell clinicians about this test when they ask? So I think some of the clinical questions that your docs are going to want to know is what viral load predicts disease? And you're like, wait a minute, I just built this fabulous test over here. I have no idea what viral load predicts disease. That's a completely different question that I didn't even, set out to answer. But that's what they're going to want to know. What level of DNA should I worry about? What level of DNA should I treat? How long should I treat? And how frequently should I test? And so we have to be able to answer these questions so that our docs know what to do with it. So how are we going to bridge this gap between our laboratory tests and what they actually need clinically? And that's where clinical studies come in. And I'm going to take you through some of the most important clinical studies done for CMV viral load. And what you're going to notice is many of them have not been published recently. These are very difficult studies to do and very expensive studies to do. And one of the reasons we don't have a CMV viral load assay that's FDA cleared is because no company has invested the tens of millions of dollars that needs to be invested to do the perfect study. And that's a problem right now. So the first question we're going to ask is what viral load correlates with disease? Or when should preemptive therapy be initiated? So this study was done in Canada by Atul Kumar. It was published in 1999, and it still remains the single most important natural history study of CMV infections in solid organ transplants. And in this study, it was liver transplants only. He enrolled 97 patients, and he followed them for 12 weeks post-transplant. Okay? He didn't use any preemptive therapy. And the only prophylactic therapy he used was if the recipient was seronegative and they received a seropositive donor. Okay? And then he watched people for 12 weeks to see who got disease and who didn't get disease. This is a study that you can appreciate would not be done today. Because of data that we know from this study, nobody would do that. But he was able to do it back then. And of his 97 patients, 61 had CMV infection. Which means 61 had a positive, either a PCR test or an antigenemia test. Okay? This is why we either use prophylactic or preemptive therapy. That's a very high number. Look how many went on to get disease. 21 of his 97 patients actually went on to get disease. But because he was able to let them go on and get disease, he was able to determine what viral load is a very good predictor of disease. So this is your ROC curve. You have one-line specificity on your x-axis and sensitivity on your y. And what you can see, if you look at any positive viral load, you're very sensitive. You pick up all your disease, but you're not very specific. Your specificity is only 50%. And as you raise the viral load that you use as your cutoff, you lose some sensitivity, but you gain quite a bit of specificity. So if you look here at 20,000 copies, and this testing was done with the Amphiphore monitor assay. All three studies I'm going to show you were all done with the monitor assay. So it takes that assay variability out for you. So if you use a monitor assay, anybody that had a viral load of greater than 20,000 had disease. Okay? So that's a high viral load clinically. And what we also learned is that a viral load between about 2,000 to 5,000 was very predictive of disease. And so I used this assay in the clinical laboratory for years. And when I would see patients on the floors, this is a trigger point that I had burned into my brain. A couple of thousand copies, you get above that, and there's too much of a high risk for disease. So this is just to show you, if you use any positive result, the sensitivity was 100%, but your specificity and positive predictive value weren't very good. And as you raise the viral load for your cutoff from 2,000 to 5,000, you got your specificity down to 86%, your sensitivity down to 86%, but you got your positive predictive value up to 64%. So I tended to settle somewhere in the 1 to 2,000 range with this assay. Now the other important paper that was published was not only is the absolute viral load important for clinical decisions, but the rate of rise. And so if you have a patient who is at 500, 600, 700 on a weekly basis, they're very different than a patient that goes from nothing to 5,000. That person is at a much higher risk of developing disease. And so the rate of increase is important. And not only is the actual number, but the pace at which the patient is getting there. So if you're going to predict disease, it really depends on the type of transplant. In bone marrow and solid stem cell transplant recipients, we still are very conservative, and we tend to treat any positive result. But on solid organ transplants, with the data I just showed you, particularly in liver transplant recipients, in the Amphiphore assay, you can use a cutoff of a couple of thousand, and I think that's a very good point for making decisions regarding preemptive therapy. But remember, that depends on the test, okay? That's true of the monitor test. If you develop your own assay, you need to either know how it compares to the monitor test, or you need to collect clinical data and begin to figure out what the important trigger point is. It also depends on the specimen type. Okay, all the data I'm going to show you is done on plasma specimens. The viral load in whole blood is about 0.6 to 0.7 log higher than it is in plasma. So if you're going to use a whole blood-based test, your cutoff points are going to be very, very different. And right now, because we don't have an international standard and because we don't have any great agreement of values across all these different tests, we don't have a good perspective on what cutoff could be used broadly. Every lab kind of has to establish their own cutoff. Okay, so that's what viral load is important. How long should we treat our patients? So what do we know about this? There's a nice paper done at the Mayo Clinic that looked at 24 solid organ transplant recipients with CMV infections, and they gave them 14 days of IV ganciclovir. Okay? They did the viral load before therapy and after therapy. And again, we used the monitor test, or they used the monitor test. They found 8 of their 24 patients actually had relapsing infections. So at the end of the 14 days, as they continued to monitor them, they got infected again. And so the question was, can you predict, based on viral load, who is going to have a high risk of relapse? And so what they did, this is their pre-treatment viral load, and this is their post-treatment viral load. These are leukocytes, and these are plasma. So if they tested leukocytes, what they found was the pre-treatment group and the relapse group, the non-relapse group had a viral load a median viral load of 5,500. Those in the relapse group, pre-treatment, had a viral load of 80,000. So you could tell by the starting viral load who was at a high risk of relapse. And you could also tell by the ending viral load. The non-relapse group basically all cleared their DNA from leukocytes at the end of therapy. But that was not true of the group that relapsed. So if you still had detectable viral load at the end of treatment, that was a risk for relapse. Now, in plasma, it's interesting. There's a difference between the non-relapse and the relapse group before therapy, but it wasn't statistically significant. But it was very statistically significant in plasma post-treatment. So anybody who doesn't take their viral load to undetectable at the end of therapy has a substantial risk of relapse. Okay, remember, a third of these patients relapse in this study here. So that's a very important indicator. So what do? We tell clinicians, treat, because we use a plasma-based assay, don't we? You treat until it's undetectable. That's very important. And don't stop before you drive the DNA to negative in plasma. In cell-based, there are people that can have low levels of viral load in a whole blood sample and be doing fine. And so there you drive it down, but you might not take it all the way to 0. And not all the people that hang around with low viral loads of 3, 4, 500, not all of them will relapse, but you do have to monitor them very closely and keep an eye on the rate of change. So this is extremely helpful information for us. Okay, so the next question you might want to know is my patient failing therapy? I'm kind of early in my treatment course. I'm getting a little nervous here. Are they responding as they should? Well, what is the kinetics of CMV DNA in plasma? And so this is another study that Atul Humor published looking at viral load kinetics in relapsing infections. So he had 52 organ transplant recipients with CMV disease, and again, 12 of them, or 23%, had recurrent disease. And so what he did was a little bit different. He went and determined the time of clearance of DNA from plasma in his relapsers compared to his non-relapsers. And then he also determined the half-life of CMV in his relapsers and his non-relapsers. So in the people that relapsed, the time to clear was about 34 days. In the people that didn't relapse, the time to clear was 17. Now that's statistically significant, but is it clinically important? Because how long it takes you to clear your DNA depends on how high your viral load is to start with. So yes, it took longer to clear, but that longer time to clear may have been because they started with a higher viral load. What's interesting is the half-life. The people who did not relapse, the people who did relapse had a viral load of about 9 days, and the people who did not relapse had a viral load of about 3 days. I find that to be very helpful. So now I have a patient, I suspect CMV disease. I get a viral load and it's 35,000. I'm like, okay, we got a problem here. I start therapy. When should I test them again? Well, what clinicians can't help themselves, they want to test the next day. It's like, that's what I do with blood sugar. Let's just test the next day. Well, with a half-life of somewhere between 3 and 7 days, say, you don't want to do that. And so what we've taught our clinicians is wait 5 days. I know it's going to be hard for you. Pace around. Maybe if you order it to be drawn in 5 days, that would make you feel better. But don't get a viral load the next day. You must get a viral load the day you start therapy, but then don't try again until 5 days, and then wait another 5 days. Sometimes that 5-day level is not really that much different, but when you get out to 10 days is when you start to see the dramatic drop in viral load. And these are just some examples. This is a patient who post-transplant had very low viral load. These black circles here are an antigen assay, the antigenemia assay, and these open circles here are the PCR. And what you see is this person was negative post-transplant for quite a few weeks, and then went from nothing to a viral load of 10,000 in 1 week. So what I've told you is we got a problem here because, one, the rate of rise was very rapid, and two, the absolute level was quite high. And in fact, this person had symptomatic disease right here. So they were started on ganciclovir, and you can see the first one after therapy actually went up. I hate when that happens, but it does happen. And then as they continued to test, you could see the viral load went down. It went down completely to undetectable in a relatively short period of time. And then therapy was stopped, and there was a little bump in viral load, but it was more in about the 1,200 range, and not clinically significant. So this is a good, nice response. This is someone where it's not so nice. They start out negative, and in a 1 week period, they go from nothing up to a viral load of 10,000. And then this person gets started on ganciclovir, and you can see the viral load isn't going anywhere. And this is a very bad sign. They were finally out here switched to foscarnet. The viral load came down, but then quickly came back up. This we found out, in retrospect, was ganciclovir-resistant virus, and this virus out here was both ganciclovir and foscarnet resistant. So watching the trends is extremely helpful. And now we know that once we got out to about here is when you really need to intensify therapy. And you can now do resistance testing for ganciclovir right out of a plasma specimen. As long as the viral load is 3,000-5,000, you can send that off for sequencing, and people will look for the mutations in either the UL97 gene or the polymerase gene. So we have a lot more tools at our hands. Back in the battle days, when this patient was being followed, you would wait 6 weeks for a phenotypic resistance testing for CMV. And, 6 weeks, give me a break. I have to make many decisions before that test is going to come back. So our toolbox is a little better than it used to be. Okay, so what have we learned about failing therapy? Plasma load half-life is somewhere between 3 and 8 days. Time to clearance will depend on what viral load you start with, but you really have to be patient and test 5 to 7 days into therapy. And this is really influenced by the half-life of the virus, but also the reproducibility of your assay. A viral load that doesn't decline or has a delay in decline or comes down and then comes back up, these should all be concerns for you that there is resistant virus. And when a clinician calls and says, I need resistance testing, have them talk you through why they need it. And if it fits into these categories here, it is an important reason to send resistance testing. So how do we actually use viral load testing at Emory? And it really depends on who you talk to. Each of the services is different. But our bone marrow group, as I said, they monitor their patients very closely on a weekly basis for into the first 3-4 months after transplant, and then they go to every other week. And often they monitor them for a year or longer, depending on whether they have graft versus host disease or a variety of other factors. They rely on screening and preemptive therapy, and they've done very well with this. Our assay is sensitive, and they're not finding much disease, and they're having even less disease that leads to mortality. It's very rare now for us to have someone die of CMV disease. Any positive result is treated until the PCR is negative times 2, and at least they get 2 weeks. If it takes longer to clear than 2 weeks, then they'll get longer therapy. Then they'll give them maintenance therapy for a few weeks. We're seeing more late disease. As we aggressively treat disease early, we're seeing that in some patients, we're seeing disease at a time when it wasn't really on our radar screen, farther after transplant. Our renal group prophylaxis, because we are a regional transplant center and our patients leave and go all over Georgia and parts of Florida and Alabama. And so getting good follow-up and getting good follow-up with the same viral load assay is not always easy. So they prophylax and don't routinely monitor people. Our liver group does monitor. If the donor is positive and the recipient's negative, they get prophylactic therapy for reasons that we showed earlier. And anyone who's recipient who's seropositive will be monitored weekly, and if it's positive, given preemptive therapy. Our lung group uses prophylactic therapy, but once they finish their prophylactic therapy, they use the viral load test to monitor them later. So it's an interesting mix and match amongst our groups. So what can we say? What have we learned here? Well, one, CMV is an important pathogen in transplant recipient, and clinical management is actually quite complex. The approach to assay verification we've gone through, the importance of understanding the reproducibility of your assay so you can determine important changes in viral load, and you can help educate clinicians. You're sweating over something that is within the variability of the assay, and that's very important information to know. The role of log transforming data, it helps physicians not overinterpret small changes in viral load. Know the agreement when you change tests. This is critical. When we went on to the data that I showed you for this assay was with a manual extraction method. If you use an automated extraction method, what we saw was the values of our new assay were on average 1/3 the values of our old assay. And that made people really anxious. And what ended up happening, plus it was a little bit more sensitive, and what ended up happening is we had all these low positive results that they never used to see before. And so I was in the hospital on service one day, and I was accosted by one of my renal transplant colleagues telling me that I ruined her life because the old assay we used wasn't that sensitive, and they weren't getting all this information. They weren't getting all these low positives, and she had no idea what to do with the information. And so what we've done now is we've collected a series of patients, I think we have about 50 now, that have these low positive results, some did not get therapy, some did get therapy, and we're going now through record review to figure out what happens to these patients. And is there a way we can tell who's at risk and who's not? Now, an interesting question is, what is it going to take to get a test through the FDA? And I am in conversations with actually people at the FDA and various companies trying to address this question, and it's an extremely difficult and likely expensive clinical trial to have to do. And it's going to be very challenging. I'm more optimistic now than I was a couple of years ago, but we're still not there yet. So viral load testing for CMV plays a key role in managing transplant recipients. It can predict disease. It can not only the viral load, but the rate of viral load. The rate of decline in viral load is extremely important. Viral load level at the end of therapy is very important. These are all things we've learned from clinical trials. Specimen type we talked about plays a role. And so what it allows me to do, this viral load assay, is really manage a patient very closely. I can look at sequential viral loads and predict how they're responding. Do I need to intensify therapy? If I do a viral load on day 5 and on day 10 and it hasn't budged, not only am I going to send out resistance testing, but I'm probably going to add phoscarnet or add immunoglobulin. So it gives you a lot of options on how to manage your patients. The important thing that you have to understand from a laboratory perspective, it is a whole lot easier to design a reliable CMV viral load test than it is to determine its clinical utility. Okay, we have the easy job. The difficult job is on the clinical side, and that's why partnering with your clinical colleagues is really necessary for us to figure out the best way to use and apply these assays. Now, I just want to talk briefly about where we're going in the future. I think for CMV, there is a tremendous need for an international standard. And I was at a meeting in the UK in June where the whole concept of an international standard was discussed. And so I was at the National Institute of Biological Standards and Controls, which is one of 2 organizations that the WHO has contracted to make international standards. They're the ones that made the hepatitis C, the hepatitis B, and the HIV international standards. So a project is now underway to produce an international standard. And what they'll probably do is culture up virus and either purify nucleocapsids or maybe just use a supernatant from a viral culture. And they'll dilute this out, and they will send it out. There'll be a call for laboratories around the world that do CMV testing to give them an aliquot and say, do the testing. They'll take all those results back, and they'll throw away the outliers, and they will determine a viral load. It is by no means probably a true viral load because anybody who wants to participate can participate, but at least it becomes a value that we will all agree upon. And once we have that international standard, then, as I said, companies can make secondary standards and we can all get our viral load assays in much better agreement. And so this project is likely going to be done in the fall of 2010. And that, I think, will help move the whole field forward. I think another interesting topic is where your testing should be done. There's a lot of move towards point of care. Is CMV viral load testing something that you would consider doing point of care? And I was on an airplane with a colleague of mine that who runs our transplant infectious disease program, and I turned to him and I said, Marshall, if I could give you a point-of-care viral load test that you could do in the clinic in an hour, what would you think about that? Is that a waste of time, or would you like that? Well, of course, he loved it. And the reason he loved it is because the patient's right there. He gets the result, and he can make his management decision in real time. We run CMV viral load Monday through Fridays, okay? But still, that means you're going to have to make an extra phone call at the end of the day or the next day to touch base with your patient to figure out what you want to do with that information. So there may actually be a role for this. And you know, I think the important thing about point-of-care testing is everybody loves it. Ask a physician if they want a point-of-care test. They'll say yes to everything. They love the concept of getting the answer fast. And they don't understand the concept of reliability, accuracy, reproducibility. So your job as laboratorians is to educate them that, you know, oh yeah, there's a point-of-care test here for influenza antigen. It has a sensitivity of 45 to 55%. You like that? Well, minor detail, if it's positive, it's helpful. What are you going to do when it's negative? I don't know. I haven't really thought that far. It's your job to help them understand that, yes, it may be fast, but it may not be very good. And so my key question that you always have to ask a clinician is not if they want it, not if they need it, but whether will it impact your management. And if they can't give you a nice, coherent how this is going to impact your management, then you need to be suspicious. So I'm going to take you through, just in the last minute here, this system called Lab in a Tube by IQ. And this is a system that's in development right now. I think they've done some development tests for HIV, and we're actually supposed to work with them on CMV. And this little gadget here is a flexible little plastic tube that all the extraction of the nucleic acid and the amplification and detection occurs in this tube. Okay? So what you do is, and this is a little, you can tell their marketing people went a little wild, because it's unlikely we're going to stick someone's finger on the top of this tube. It's more likely we're going to collect it in the blood and collect it in the tube and enter it into here. But you add your specimen directly to this tube, so right into this section here. And then you scan the barcode, and you put it in this adorable little instrument here. And in an hour, you have a viral load. So it's really an amazing gadget. There are multiple companies out there that have these types of instruments and these types of methods in development for molecular testing. And I think you're going to see a real shift in the next 3 to 5 years, if it even takes that long, because for those of you in this field, you know that the Gene Expert by Cepheid can give you a result on an RNA virus in about 2.5 hours and on a DNA target in about an hour. And so we're going to all of a sudden enter this world where molecular results are going to be available to us in a much faster manner. And your job is going to be to determine whether or not that's a good use of money or a bad use of money, because some of these tests are extremely expensive and some of the instruments are actually pretty expensive. So these are some of the interesting issues that we'll be dealing with in the upcoming years. And I think I'm done, and I will take any questions. Thank you.